Timothy syndrome is a rare genetic condition that affects the heart and several other organs. In the heart, Timothy syndrome manifests as the prolongation of the QT interval on the electrocardiogram that predisposes to irregular heartbeats and cardiac arrest. Timothy syndrome arises when a DNA mutation impairs the function of a protein called calcium channel, which is responsible for regulating the transportation of calcium ions across cell membranes in several organs of the body. Abnormalities of calcium flows across cell membranes impacts the heart's electrical activity, leading to irregular heartbeats called arrhythmias and sudden death in young individuals. Yet Timothy syndrome's extreme severity and rarity hampers researchers' ability to understand the disease's pathogenic mechanisms, stratify risk, or develop new therapies. For this reason, Professor Priori from the Maugery Research Hospital at the University of Pavia and the Research Institute CNIC in Madrid, in collaboration with Avantea in Cremona, are working to study the disease in genetically modified pigs. The researchers used a complex technique called CRISPR-Cas9 to introduce into a pig embryo the same mutation of patients with Timothy syndrome. The embryo is then implanted back into the sow, giving birth to piglets with the Timothy syndrome mutation. Professor Priori has shown that the genetically modified pigs have the same clinical manifestations that Timothy syndrome has in patients, including alterations in electrocardiograms and the development of life-threatening ventricular arrhythmias that can lead to sudden death. With the help of these Timothy syndrome pigs, Professor Priori and her team are developing treatments including novel antiarrhythmic drugs and potentially curative innovative gene therapies. Funded by the European Research Council, the team is pushing forward to bring this research to a first-in-human clinical trial.